In the oldest forests, each tree is part of a secret underground web, living threads, finer than a human hair, weaving root to root for kilometers beneath your feet. This hidden network trades resources and broadcasts warnings, shaping which trees survive and which perish. But what really flows through these mushroom-tied connections, and how much do trees truly share? The answer will upend everything you thought you knew about life in the woods. The alliance between trees and fungi runs deeper than most people realize. In 1885, the botanist Albert Frank named the partnership mycorrhiza, which means fungus root. Under the forest floor, fungal threads wrap around the tips of tree roots, forming a living sheath called a mantle. Early micrographs from the 20th century revealed these mantles as dense, tangled coats invisible to the naked eye. This partnership is built on trade. Trees produce sugars through photosynthesis, feeding the fungi. In return, fungi gather minerals and water from the soil, nutrients that roots alone could not reach. This exchange shapes the health and growth of both partners. The network that emerges is not a simple pipeline, it is a marketplace where carbon, nitrogen, and phosphorus pass between species, guided by need and opportunity. Every connection is a negotiation, every transfer a balance of survival. A single teaspoon of forest soil can hold more than 5 kilometers of fungal threads. These hyphae, each thinner than a human hair, weave an invisible web through the earth, branching and spreading in every direction. Their numbers surge and fade with the seasons. After heavy autumn rains, mushrooms erupt above ground, just a fleeting sign of the vast hidden network below. Most of the time, the true extent of these threads remains concealed, packed tightly around roots and stones. The density is staggering. In 1 square meter of healthy woodland, the combined length of hyphae stretches far enough to circle the globe. Yet because these filaments are so fine and densely packed, watching their movement or the flow of nutrients through them defies ordinary observation. To trace the living currents within this underground maze, scientists must turn to special markers and tracers, making the invisible visible. Resource flows through the mycorrhizal network are not just theoretical, they are measurable and at times surprisingly swift. In controlled field experiments, up to 4% of a donor tree's newly fixed carbon can reach a neighboring sapling within 48 hours. Nitrogen tracked using the stable isotope 15N leaves its signature in recipient leaves with enrichments on the order of 2 to 3 parts per thousand above background. Phosphorus, essential for growth, moves at a different pace, traveling along hyphae at rates measured in millimeters per hour. These exchanges occur continuously, forming a living current that responds to the changing needs of each plant. The numbers reveal a system where even modest transfers can mean the difference between survival and decline, especially for seedlings and shaded understory trees. In a controlled greenhouse chamber, 2 young Douglas firs stand side by side. Their roots are separated by a fine mesh, just wide enough for fungal threads, but too narrow for roots to cross. Above one tree, researchers release a pulse of carbon dioxide enriched with a rare isotope, marking every sugar molecule the tree produces. Over 24 hours, the tracer appears in the neighboring seedling, but only when the fungal network is intact. When the mesh is replaced with a tighter weave that blocks hyphae, the transfer stops. Suzanne Simard describes the moment. "We saw the label move only when the fungus could bridge the gap. It was the living network making the connection. The uptake curve rises sharply in the connected treatment, and it flatlines when the network is severed. In this experiment, the invisible web becomes visible proof. Carbon flows only through the fungal bridge. At the microscopic level, the proof of these hidden pathways becomes undeniable. Confocal laser scanning microscopes reveal halos of fluorescent tracers glowing around root cells where fungal hyphae have delivered their cargo. Each bright ring maps the journey of labeled phosphorus or carbon, tracing the exact route from one tree's root to another. NanoSIMS imaging goes deeper still, pinpointing isotope hotspots with submicron precision. These maps show clusters of 15N or 13C not only in the hyphae themselves, but inside the recipient plant's tissues. Layer by layer, these images confirm a living network, measurable, mapped, and real. The underground web that links trees is not always a force for good. Alongside nutrients and water, fungal threads can carry threats. In one experiment, scientists traced the spread of a root rot fungus across a common mycorrhizal network, watching as infection moved from one tree to its neighbors through shared hyphae. The same connections that support young seedlings can also drain them of carbon when mature trees dominate the exchange. In dense stands, saplings linked to powerful hubs may lose more sugars than they gain, stunting their growth. These networks can become highways for help and harm, shaping who thrives and who falters beneath the forest floor. Protecting the underground network begins with choices made above ground. In forests managed with care, patches of old trees are left standing, their roots and fungal partners undisturbed. This approach, known as retention forestry, helps maintain the living web and connectivity beneath the soil. Studies show that partial cut systems can preserve up to 70% of hyphal continuity, compared to only 20% in areas that have been clear-cut. Avoiding heavy machinery and minimizing soil compaction also keep the network intact. A forest steward reflects. When we leave legacy trees and treat the soil gently, we are not just saving timber, we are safeguarding the entire web of life that supports the next generation. Forests aren't just collections of trees, they are living networks, shaping each other's fate in real time. In every route, tomorrow's forests are being negotiated. What role will we play?